My name is David Nwobodo, and I'll be presenting on enhancing antibacterial compounds biosynthesis in endophytic Classidioploidea tuberome and Coculture lunata through co-cultivation. My outline: endophytic fungi are fungi that reside within plant tissues without causing any harm to their hosts. Rather, some of them have been said to protect the host in hostile conditions by the production of secondary metabolites. The search for novel bioactive natural compounds is gradually shifting from the traditional methods that we all know to several advanced techniques that involves engineering strains to influence regulatory systems that governs the expression of secondary metabolites in biological systems. One of such techniques is the coculture technique. Infectious diseases remain one of the leading causes of mortality in the world. The bioprospection of endophytic fungi over the years have offered significant promise in the discovery of secondary metabolites or agents for the treatment of these infectious diseases. However, the biosynthetic pathways that are responsible for secondary metabolites have genetic basis for their production, but the expression of these genes are not well observed in arsenic condition when isolated in the laboratory. Aim and objective of the study: materials and methods involved in the study, other materials and equipment used in the study. The plant materials used in the study were collected, sterilized, and then cultured on potato distress agar. Then the organisms grew and were purified to obtain the pure isolates. The pure isolates, the two organisms were used to culture, to ferment, they were fermented individually and then used together in the co-culture study. Fermentation was carried out using rice medium as a substrate for 21 days, after which the crude extract was concentrated and extracted using ethyl acetate as a solvent, and then tested for antibacterial activity and the GCMS analysis to identify the bioactive compounds. Organism A, L. tuberome, was isolated from Cola acuminata, while organism B, Coculture lunata, was isolated from Bambusa vulgaris. Both organisms have been reported to be associated as endophytic fungi of different plant parts in time past by other researchers. The extract of L. tuberome inhibited only Staphylococcus aureus, while that of C. lunata inhibited Staphylococcus aureus, Bacillus subtilis, and Pseudomonas aeruginosa. The co-culture extract only inhibited Staphylococcus aureus, however, at a higher IZD value. This might imply that the agents that inhibited Staphylococcus aureus or agents that inhibit Staphylococcus aureus were enhanced, while agents that inhibit Bacillus and Pseudomonas were antagonized in the co-culture experiment. Gentamicin served as a positive control in this study. 13 compounds were identified in the L. tuberome extract, while 14 compounds were identified in the C. lunata and 11 in the co-culture extract. Compounds highlighted in light brown are those that were antagonized, while those that are highlighted in light blue are those that were enhanced. In the co-culture experiment, 5 new compounds that were not identified in the monocultures were identified, and these compounds are compound number 5, 8, 9, 18, and 21. E. coli was resistant to all extracts in this study, both the monoculture and the co-culture. This could be as a result of the outer membrane of this organism made up of lipopolysaccharides that have been reported severally to protect the cell from external agents. The majority of the compounds identified here are essential oils, which may denote the antimicrobial activity observed against susceptible bacterial strains. These compounds have also been reported to be produced by other endophytic fungi in other studies and also shown to have antibacterial activity. For the co-culture extract, the most abundant compound is an antimicrobial agent called pharmacin. Statistically, the concentration of this compound was highly significant when compared to that of other compounds identified in the co-culture. This means that this compound was not identified by chance, rather was produced as a result of the interaction between both organisms used in the co-culture study. Conclusion and recommendations, selected references. Thank you.